All right. Good morning, good afternoon, good evening, depending on where you are, all beer family members. Welcome to day 3, our oral presenters session. We are once again delighted to have you join us. I assume as we continue, more oral presenters will join. So this is now the day for the oral presenters to shine. We invited questions for specific oral presenters through the Q&A form, which we already compiled and we have. So the way we are going to proceed today is we are going to give each oral presenter about a minute elevator pitch opportunity to talk about their presentations. Your presentations have already been posted and have already been watched. So you'll be basically giving a brief, very short summary of your talks. And then we'll invite questions from the attendees through the chat. So if you are in the attendees room, as the oral presenters are pitching their talks, feel free to send your questions through the chat. We'll compile them, add them to the list, and then we'll combine your questions sent through the chat and the questions we got from the Q&A form and direct them to each individual speaker, and then we'll wait for their answers. So once again, I'd like to welcome you. Perhaps, Sarah, would you be interested in charge of designating the speakers, because my screen, the pictures keep moving. So who should start first?
Yeah, I guess I'll go from left to right from what I see. And the first person is actually Demond. So Demond, do you have the joy of giving a brief introduction?
In the meantime, perhaps all other speakers, if you could mute yourselves, that way we'll only get to hear the speaker. Thank you again.
Can everyone hear me? So my name is Damon Hanley. Just really quickly, I'm a PhD student here at The Ohio State University studying biostatistics. And the presentation that I gave, it focused on using gene expression data to build a neural network model. And that way we can predict whether or not a person has coronary artery disease in non-diabetic patients. And the data that we used for that model was based off of a previous study, which developed a model to test patients for coronary artery disease. And the model that they built was based on ridge regression. So we built a neural network model and we tested it against, we tested that model, well, we compared that model against the previous ridge regression model, and we also compared that model to other machine learning methods such as support vector machines and random forest classification. I worked on this project under the direction of Dr. Achman.
Awesome, thank you. And then next on my list is Brandon Stigler.
Hi, good morning, everybody. I guess you all can hear me. So I'm at Southern Methodist University. My research area is algebraic geometry applied to mostly the network inference for gene regulatory networks. And what I presented today, or I guess this week, and was looking at an algebraic framework that we've developed for doing data identification or design of experiments and model selection. And so it's basically what we do is that we produce bases for models. Presentation was, do we have an upper bound on the number of models that would be associated to a data set in the reverse engineering process? So thank you.
Very cool. Next on my list is David Chan.
Hey, hello everyone. I actually gave two brief talks. One dealt with looking at support networks in college students, and in particular, those networks that affect their education and mental health. And we found these special people in some of these networks, which we called prime supporters. And in the talk, I talked about how these prime supporters affect both the student's education and mental health. And the other talk I gave dealt with a difference equation model, which was trying to mimic seizures, epileptic seizures in the brain. And so we found some interesting dynamics in that model. In particular, we found these types of, they weren't periodic orbits or almost periodic orbits, but they were what we're calling strange periodic orbits that sort of followed a periodic orbit and then got reset, but didn't reset to where it was. So it's interesting dynamics. So those are the two things that I talked about. Thanks.
Awesome, thank you. And then I just to give a brief update. If I lose power, my lights have just started flashing. Someone else will have to take over, but hopefully it's good. Next on my list is Kelly Butch.
Hi there. My name is Kelly Book, and I'm a PhD student at the University of Tennessee in Knoxville. My dissertation work is in forest tree diseases and how an individual tree's response to disease can affect the spread of the disease at the forest level. So I'm specifically looking at right now the role of basal sprouting in vector-borne tree diseases, and I'm using laurel wilt as a case study. Laurel wilt is a fungal vector-borne disease with devastating effects in the southeastern United States. So I'm looking at the role of the basal sprouting and how that helps population maintenance, but then also kind of keeps the epidemic around for longer.
Oh, very cool. Next on my list is the Julien Q. Is that right?
Almost. So hello everyone, I'm Jolene and my last name is Chu. So
Sorry about that.
yeah, I can recognize those. So in my talk, I introduced our new model, which is a stage progression model of compartmental model for modeling the invasive non-typhus salmonella epidemic in the sub-Saharan Africa. So we come up with this compartmental model, and where we have 4 population cohorts and 8 infectious disease. So it's like a monster system. And we derived the basic reproduction number, and we gained some quite interesting results on how we understand the contributes of each compartments towards this number. Specifically, we include this asymptomatic stage of the disease, so we can see how that affects the R0 calculations. And yeah, I guess that's pretty much it. Yeah. Thanks.
Very cool. Next on my list is Anna Sisk.
Hi, yeah, I'm Anna Sisk. I'm a PhD student at the University of Tennessee in Knoxville. And my presentation was on applying principles from epidemiology and immunology to violence. So looking at how we can use mathematical epidemiology and immunology models to study how violence spreads. And I was particularly looking at using mathematical epidemiology models to look at how violence spreads in a community.
That's awesome. I guess maybe I shouldn't say awesome, but it's a cool idea. Next on my list is Deborah Smita.
How do we determine?
Hello everyone, I am Devoshmita Mukherjee from India. I am an assistant professor here, and I have submitted my work on atherosclerotic modeling. So atherosclerosis is a chronic inflammatory disease. It occurs due to plaque accumulation in the arterial wall, and I tried to formulate that in mathematical terms, and my model consists of 10 ordinary differential equations, nonlinear model, and then I have applied numerical analysis on that model, and then I tried to define the numerical outputs in biological terms. And there I have found few significant bifurcating parameters, and I hope that in future some expert in clinical department, they can take the model and generate some therapeutic strategies for a group of patients. And thank you all.
Thank you. Next on my list is Tugba Akman.
Hello, everyone. I'm from Turkey. I work for University of Turkish Aeronautical Association as an associate professor. My talk was on pancreatic cancer modeling. Actually, we considered an existing mathematical model, and we fit the data to obtain just a few values of the parameters, which we taught them as patient-specific. And then we extended the model with cancer stem cells, and we just tried to understand the progression of this disease. But actually, the tumor cells increased too much, so that we cannot directly catch the reason behind of this increase. And we just try to understand the effect of chemotherapy and immunotherapy with the use of the data. But since this type, the pancreatic cancer is quite aggressive, chemotherapy is not, working good. Therefore, from now on, we will be concentrating on the targeted chemotherapy so that hopefully we can decrease the number of tumor cells in the long run and increase the survival time. Thanks.
Thank you. Next on my list is Sharon Buick.
Hi. So I built an online tool that could be useful for predicting the number of students returning to campus carrying a COVID infection. And to do that, we combined IHME models. So that gave us the predictive amount of sort of COVID spread in any one state. And then we built a database of 1,440 universities and colleges across North America that told us the breakdown of how many students were returning from each state. And so we combined the predictive model with the sort of breakdown of students across a campus so that people could pick their school, they could pick the return date, they could pick a couple of disease parameters like how long people were infectious and how long the incubation stage was, and then predict how many students would be coming back, on the plane first day carrying the disease.
Very cool. Next is Madeline B.
Hi, so my name is Madeline Brown. I'm an undergraduate student at Scripps College. And so my research was focused in knot theory. So if knots have homeomorphic complements, then they're isotopic. So what that means is like if there's a continuous mapping from the space surrounding a knot to another, then those two knots are equivalent. But the same isn't necessarily true for links. And so what our research focused on was two types of hyperbolic links and determining a way to see if they're isotopic, if they have homeomorphic complements. And so we did that by finding their graphical representations and built an algorithm to find their linking matrices directly from the graphical representations. And what's kind of neat about that algorithm is it still contains the topological information from the links themselves.
Well, that's very awesome. Next up is Glenn Letter.
Hi, I'm an emeritus professor at the University of Nebraska. My video is on an educational module that I wrote to teach students about COVID-19 using MATLAB or R. The module consists of programs and questions for students to address. Only minimal coding is required. And the questions are things like assessing claims made by public figures and predicting the effects of interventions like masks, distancing, and contact tracing.
That's awesome and really relevant right now. So great. Next up is Dimitri.
Hello, thank you, Sarah. Thank you, everybody. My name is Dmitry Kondrashov. I'm instructional faculty at the University of Chicago, and I gave a presentation on changing the design of a course in light of remote teaching. So it was a course on quantitative modeling, and I made some modifications besides teaching online. So I got rid of exams. I instituted very flexible grading policies, and I wanted to compare the performance of students as well as their subjective responses to surveys pre and post the course, compare them with the normal year before. So that's essentially what we did. And the main takeaways that spoiling the punchline are students like not having pressure of exams. There is no real evidence that we could see that they actually learned substantially less in the online learning. And finally, that students prefer and find more useful having non-asynchronous resources as opposed to being required to sit through synchronous lectures. So thank you very much.
Thank you. Next up is Sarah Clifton.
Hi, I'm an assistant professor at St. Olaf College in Minnesota. We looked at the effectiveness of antibiotics in treating bacterial infections in humans when there are naturally present bacteriophages in the system, one type of bacteriophage that kills bacteria and one that does not. And the main takeaway from our research is that if the bacteria are resistant to antibiotics, the infection can actually get worse if you treat with antibiotics.
Oh, really interesting. Thank you. Next up is it Nyuk Nguyen?
Hi, everyone. I'm here to have a joint talk with Dr. Feihan Azizi, and she actually the first one in the talk. And so, well, I think it would be better if she start with the introduction.
Hello, everyone. I am a mathematician, and in this work, we studied PK/PD models in discrete time. So our model contains difference equations and fractional difference equations. And we calculate the parameters, we did some statistics, we did some coding, and as a conclusion, we can say that our models, discrete and discrete fractional, do good job, as good as continuous models. Not, you want to add something?
Sure, TTC. I would like to also bring to the fact that like Dr. TTC point out in the talk that the discrete models is kind of easier to work with in the sense that it's more intuitive and it doesn't require any numerical approximations.
Yeah, that's awesome. Next up is Emily Horton.
Okay. Hi, my name is Emily Horton. I'm a PhD student at Virginia Commonwealth University studying applied mathematics, and I presented a stochastic multi-host model for West Nile virus, specifically in zoonotic West Nile virus, so between birds which function as the host and mosquitoes which function as vectors. We wanted to present two species in our model because we were able to investigate some really interesting dynamics when the two species of hosts might exhibit differing competence or maybe differing number of bites or their ability to transmit the disease.
Oh, very cool. Next up I have Prachi Das.
Yeah, hi, I'm Prachi. I'm a PhD student at North Carolina State University, and the presentation that I gave was about a model that we worked on that looked at how pathogen mutation can affect the emergence of vector-borne infections, so specifically mosquito-borne diseases. And this was like motivated by the fact that there has been reemergence of infections like chikungunya, where a mutation in the virus enabled a more efficient spread of the virus by a secondary vector versus a primary vector. So you've had spread to regions that were previously non-endemic. So we basically wanted to use this like branching process theory-based model and incorporate mutation into it and see how it would affect the probability of infection emergence.
Oh, cool. Next up is Paul Hurtado.
Sorry about that. Hi, everybody. My name is Paul Hurtado. I'm an assistant professor in the Department of Mathematics and Statistics at the University of Nevada, Reno. I'm also faculty in our interdisciplinary PhD program in ecology, evolution, and conservation biology. And the work I talked about is some work I've been doing the past year or two, looking at generalizing the linear chain trick. And a lot of ODE models have these sort of simplistic implicit assumptions built into them, and or they're derived based upon sort of rules of thumb that are sort of founded on these sort of simplistic assumptions. And so we wanted to go through and see how much we could generalize what's called the linear chain trick, which is basically, if you've got a state like an infectious period in a disease model, you can replace that with a linear chain of substates. And usually when we write down equations for models like that, we're implicitly assuming that individuals spend an exponentially distributed amount of time in that state. And if you've ever looked at disease data, that might not be the best choice. So the linear chain trick lets you use a gamma distribution because sums of exponentials is gamma. So it turns out the sort of natural way to think about mean field ODE models in this sense is to think about not just gamma distributions as the distributions for the time it takes to passage through a series of substates. You can actually write down a series of substates that are much more like the transient states in a Markov chain, where you kind of bounce around among those different substates. And we have matrix exponential distributions that describe what the distribution of time is it takes to passage through states in that manner. And it turns out the matrix and vector you use to parameterize those matrix distributions comes straight out of Markov chain theory. And there's like a one equation way of writing down your mean field ODEs based upon that matrix and vector, which allows you to make these sort of underlying model assumptions in Markov chain land. And then without doing any complicated model derivation, just write out what would otherwise look like pretty complicated mean field ODE models looks like. So if that sounded complicated, my talk is about how it's not. Basically, there's some shortcuts for going through and doing this, and there's some real benefits to sort of generalizing these models in this way analytically. So you can go through and do things like calculate R naught in ways where interpreting the results of that draw really heavily from Markov chain theory and renewal equation theory. And I think really adds a lot to the analytical tools we have for ODEs, sort of by using this generalized linear chain trick as the bridge to pull over Markov chain theory tools to do that. So anyway, I also plugged that I've got a PhD position open. If anybody's aware of any really good students that are interested in math bio, I'll put links in the chat there for more on the research as well as that position. So thank you guys.
Thank you. And I just want to make a brief announcement to all the panelists. I put a little note in the chat, but some of our panelists have been responding to just the panelists instead of all the attendees. So just make sure that you're responding to both if you're trying to answer the public. Next up, I have Frances. I think it's Aponte, but I can't say the entire last name.
Hi, my name is Frances Aponte. I'm a senior undergraduate student at the University of Puerto Rico at Cayey. My work is about mathematical models for sexually transmitted diseases, specifically chlamydia and trichomoniasis, that are both epidemics in the sexually active population. From between 15 to 35 years old in Puerto Rico. So I was particularly looking for interventions that could be applied to the models to decrease or even eradicate the disease, like was the case of Donovanosis in Australia, another STD. After applying an eradication plan that consisted of isolation, guarding fully completion of the treatment, sexual abstinence simultaneously. Thank you.
Thank you. Next up, I have Carlos B.
Yeah, hello everybody. My name is Carlos Bustamante. I'm a first year PhD student at Arizona State University. And my work is about the transmission dynamics of COVID-19 outbreak in provinces of Ecuador, which is where I am from. So the dynamics of the outbreak has been observed quite different depending on the province being analyzed, with high reported prevalence in some low population density provinces. So in this study, we aim to understand the variations in outbreaks between provinces and provide assistance in essential preparedness planning in order to respond effectively to ongoing COVID-19 outbreaks. So we created a compartmental model where we implemented most of the interventions that were that were currently implemented, like quarantine, isolation, hospitalization, and ICU. And we estimated like the quarantine rate, the critical quarantine rate that we wanted to achieve in order to overwhelm the hospital, in order to not overwhelm the hospitals. And also we estimated the basic reproductive number for each province. Thank you.
Very cool. Next up, I have Paul Valle.
Thank you. Hello, everyone. My name is Paul Valle. I'm a Mexican researcher from the Tijuana Institute of Technology, what you can see behind me in the picture. We are very happy that our work was considered in this symposium, me and my students. My work was focused on a model of a leukemia model, and we combined nonlinear theories of the localization of compact invariant sets method, a Lyapunov stability theory to derive sufficient conditions to ensure tumor eradication. And we combined these conditions with numerical simulations in order to provide an insight on how we can apply an immunotherapy treatment to eliminate this non-solid tumor over time. This is our work, and another friend is right here right now that's going to talk about our work in our institution. Thank you very much.
Thank you. Next up, I have Richard Clark.
Good afternoon, everybody. My name is Richard Clark. I'm a first-year master's student at Arizona State University. And I did some research with my colleague, Leah Darwin. And essentially what we did was we genotyped a local population for a protein that's responsible for reinfection, severe reinfection of dengue fever. And so what we did was we collected data from this population, and then we created a mathematical model to represent how severe an outbreak would be. And I'll pass that to Leah to talk about that model.
Hi, I'm Leah. I work with Richard Clark. I'm actually not a master's student, and I'm an undergraduate, and I'm a senior at Arizona State as well. And with regard to the mathematical model, basically the idea is that based on your genetics, you have differences in how susceptible you are to different forms of the disease, right? So what we did is we took a basic compartmental model and we split it up based on individuals and their genotype. And then we also looked at how changing their genotype may change how they move from susceptible to infected. And we got some cool results looking at different populations and their distribution of these genotypes, including Tempe, which we sampled ourselves. So it's some pretty exciting work, and we're open to questions if anyone has any.
Thank you. Next up, I have Eric Numfer.
I'm Eric Numfor. I'm an associate professor at Augusta University. Our model is on malaria HIV/AIDS co-infection, and we formulated a model with SIR type model for malaria in humans and SEI type model for malaria in mosquitoes, incorporating bed nets and malaria treatment. We established the existence of boundary equilibria and also the existence of a backward bifurcation, which tells us that the condition that when R0 is less than 1, the infection is eradicated, is insufficient in our model. We also formulated an optimal control problem and established a kind of characterization for bednet coverage and for malaria treatment in order to control the infection in malaria HIV/AIDS type endemic setting. Thank you.
Thank you. Next up, we have.
If I can take, so I know that this is a great group of faculty and students, and this on the chat there is lots and lots of faculty proposing assistantships, scholarships, and all that. So we will try to capture the chat for you, but I want to make sure that the students are watching these things also. Okay, this is a great information here. Thank you.
Yes, thank you, Prati. This is a great group we have here. So, and the chat's going crazy.
And if I can chime in just for 10 seconds,
In the flip booklet, the last page has emails and information of all attendees. So especially faculty who are interested in contacting any of the students, please feel free to grab their email and contact them directly after the conference. That's actually what one of the things that Beer Conference is proud of.
Thank you, Dr. Achman. Next we have Gerald Arcidi.
Yeah, hi, hello. I'm from the Philippines. I'm the faculty of the Caraga State University. And actually my group talks simple model about monitoring the hospital on COVID-19. So we do have some projections in the region where I am situated at. And then from this model, we also have some strategy based on the model, how to reduce the transmission. We do some coding, mostly in Python. Yeah, actually in Python. And I think that's it. Thank you.
Thank you. So next up we have Bismarck Oduro.
Hi, everyone. My name is Bismarck Oduro. I am a faculty member at California University of Pennsylvania in Pennsylvania. In the presentation, I talk about a mathematical model of COVID-19 among two subgroups, low risk and high risk populations with two preventive measures. Those with underlying chronic diseases and the elderly are classified as high risk individuals. And then the rest are low risk individuals. We did parameter estimation. We computed the effective reproductive number. We did contour plot and studied the contour plot. We also studied the susceptible and infectious populations for both low risk and high risk group. And now our observation was that the non-pharmaceutical intervention by the high risk individuals reduces the infection only among the high risk individuals. But the non-pharmaceutical interventions by the low risk individuals have significant reduction in both the high risk and the low risk population. So our recommendation was that those who are low risk should comply with the preventive measures, not just to protect themselves, but also to protect the high risk individuals. Thank you.
Thank you. It's so awesome how some of our research can be, it has such a great influence on the public in some ways too. Next up, we have Amit Sharma.
My greetings to all. I am Amit, working as assistant professor in a college from Punjab University, India. And in this conference, in this symposium, I'm presenting my work on COVID-19, mathematical model developed using various control parameters, that is NPI, non-pharmaceutical interventions, media awareness, all these. Presently, the paper I am presenting is a working model. My previous work is based on models using impulsive differential equations. And my group and all the members, we are trying to make this model using impulsive differential equations. And we are looking when the vaccine will come out, and the effect of vaccine will be periodic. And as in previous epidemic models, when we give vaccine, then the effect will be periodic. And maybe that immunity given by vaccine, it may be temporary or permanent. Then we are trying to make that model using impulsive differential equation and trying to study the effect of vaccine on COVID-19. So that's what we are doing now. So thank you, dear.
Thank you. Next up, we have, I think it's Andres Rios Gutierrez, but I can't see your entire last name, so.
Hello, good morning. My name is Andre Rios. I am from Colombia, and I work with Anush and Arun professors. We study and analyze a stochastic SEIR epidemic model with random perturbations. The random perturbations in the model capture the impact of stochastic variations due to external factors such as social behaviors that may impact the dynamics of the infectious disease. The analytical results are verified via numerical simulations of different scenarios in the parameters region. inference on the model parameters was carried out using the procedure that used the discrete time approximations based on the Euler-Maruyama method, match-mode likelihood technique, and empirical information. Finally, we illustrated using data from the 2003 and 2004 SARS outbreak in China, and currently we study data from Colombia for the recent SARS-CoV-19. Thank you.
Thank you. Next up, we have Maria.
Hey, I'm Gabby. Yeah, Maria's good. I have two names. Go by the second. It's weird.
Sorry.
I'm a PhD student at ASU, third year. And my talk was actually about collective defense in social groups, specifically in stingless bees. And we studied stingless bees because unlike most other bees, they have specialized bees for defense tasks. So they have this like big bees that guard the nest. And the interesting thing about them is that they have multiple mechanisms for dealing with crisis situations. So they have the specialized guards, but when they are missing, then they also have these tiny bees that are just regular workers that will go out and defend the nest if the guards are missing. So they have this interesting feedback mechanism. And what we found, we did this demographic model with differential equations about the different groups, population groups, like different tasks and switching between the tasks that they have in the nest. And we found that, interestingly, increasing the proportion of majors in the nest, so these big bees, is not always beneficial for the nest. So you would think, we're missing guards, let's produce more of them. But that could actually be not beneficial for colony size and colony growth if the guards mature too fast, so they don't contribute to other tasks in the nest, or if the minors are too sensitive to guard loss. So we basically just studied the conditions under the different defense mechanisms that they have actually helps, and that will help determine how they regulate ecologically these different defense mechanisms for thriving as a colony.
Thank you.
And just to add, Maria is also from South America, Colombia.
Yeah, I'm Colombian. Yeah.
Previous speaker was also from Andreas.
Andreas is
Colombia.
Colombian too.
And there are, and so what we are seeing is all over the world, the speakers are joining us. So thank you all.
Thank you, Anush. Good to see you.
National nature of the conference. So.
Thank you. Next we have Carlos Castillo, I think.
All right. Hi, that's me, and I apologize for not being Colombian. I'm Carlos Castillo Garso. I started in math bio and then studied some linguistics and made the switch to mathematics education. Most of what I'm interested in is identifying the skills, perspectives that and concepts that are key in developing a good mathematical biologist, and looking at how to generalize and develop those skills and perspectives in high school and undergraduate levels, so that we've got a little bit of a broader understanding of mathematical biology in the general population. Today's talk, which thanks to the wonderful staff here at BEER now has audio, is about looking at specific moves that mentors made in mentoring an undergraduate group as they were developing their research question and developing a model, and looking at what the most effective contributions that mentors made. And if any of you is interested in collaborating with me at some point in time, I feel like the age of online Zoom education makes it particularly easy for me to collect data. So if you would want to collaborate on having me study one of your classes or study some mentoring, I would be more than happy to do that.
Awesome. And then once again, if anyone has anything that they want to share in the chat, like email addresses or anything, it's the perfect place to share that. Next up, I lost my list, but I think it is Leon.
Hi, can you hear me?
Yes.
Okay. Hello, everybody. My name is Leon Ariola, and I'm from the University of Wisconsin-Whitewater, and I'm a professor of mathematics, and I'm also a black sheep because I was also a professor of physics there. So I feel kind of out of the normal way of approaching.
And
Oh, wow,
Leon is not from Colombia either.
No, no. New Jersey.
He looks like Colombian though.
Well, anyway, my talk, which has been in a series of probably several years now, I approach things from a completely different point of view. If you look at standard models, there are ODEs, PDEs, maybe integral equations, maybe difference equations, integral differential equations, etc. And so I look at that as sort of a macroscopic viewpoint. And then you could look at the discrete agent modeling, which is just horribly computationally difficult. And so I'm somewhere like Goldilocks soup, someplace in the middle, and I'm approaching things from the quantum mechanics point of view. And what happened is there's some very strange results that happen. One of the nice things about the quantum approach is that when you kind of smear it out and you look at expected values, it turns out that you get the same results that you do with normal ODE models, PDE models. So I can actually derive the logistic equation from first principles rather than making assumptions. But what has happened is some very peculiar events, and it has to do with something called quantum tunneling of probabilities. So if you take my talk was just a simple SI model, and it agrees with the normal equilibrium, but it predicts that once the infection is introduced, it never goes away. And when I first saw this, I was looking at a very simple single species population dynamics model. And it's like you toss a coin and 46% of the time it comes up heads and 46 it comes up tails. And I'm like, somebody's cheating. Where did the probabilities go? And that's what turns out is the quantum tunneling effects of probabilities. And I'm also working on trying to figure out how to wrangle R naught out of from the quantum approach. And also it might help us in looking for what are called black swan events. That is events that are very, very unlikely, but they have serious consequences. Anyway, thank you.
Thank you. I think the last one we have up is MD. If I miss anybody, please let me know in the chat. But MD, can you hear us?
Is there an audio issue?
Or possibly an audio issue.
Okay, in this case, perhaps perhaps we start with the questions, since there are lots of questions, and if MD can fix his audio issue, we can have him give his elevator pitch shortly. All right, so once again, congratulations to all, and I'm really impressed with all these exciting and really great work that you have submitted. I'll try to filter through the questions that we received. The first question that I see here is for Bismarck Oduro. Bismarck, if you can unmute yourself. The question to you is, your no intervention value of R naught seems to be about 2.5, which is somewhat lower than most reports. Where did you get your parameter values?
Thank you very much. We did parameter estimation using data from South Africa and Brazil. So the parameter values were obtained through model fitting.
Thank you, Bismarck. The next question goes to our most favorite beer participant today, Glenn Leder. The question to Glenn is, what's the background required for students coming into your class for this project?
Thanks. Nobody's ever called me the most favorite of anything in any context before. The background for the MATLAB R version, the background in my MATLAB R version is just basic differential equations. There's also an Excel version that requires almost no background, and which we've used successfully in a Math for Liberal Arts class. And there's no programming background required for the MATLAB R version. The programs are written so that there's very minimal changes that students need to be able to make to modify for different scenarios.
Thank you, Glenn. Thank you very much. Once again, nice to see you here. Dimitri, there is a question for you. And the question says, it was interesting that your students said they liked asynchronous when we hear that they want to synchronous for the interactions, but it sounds like your synchronous were lectures and not interactions. Is that true?
Thanks, OJ. I try to make the synchronous classes interactive, but with a class of 200 students, that was a little challenging. So I did in-class polling. I did a lot of question and answer time and demonstrations. But yeah, but it wasn't fully interactive. Let's put it that way.
All right, thank you, Dimitri.
Before you continue, Dr. Achman, there's another speaker here that just joined us, Diana. I don't know if she with us in this room.
Joanna Wares? Is
no,
that
Diana.
No. Diana, hi. Hi, everyone. Hello there. I'm from Tijuana, Mexico, and I'm a Mexican researcher at Instituto Tecnológico de Tijuana. The work that we are very interested on is on studying the ordinary differential equations, describing in diabetes schemes, also in the presence of nonlinear behavior. Therefore, the localizing method, which is an algebraic strategy, is very useful to us in order to define upper bounds, also attractiveness to a specific equilibrium point. And therefore, since the models are non-linear, means that Lyapunov theory can be applied. So these models are very tricky, and we are interested because diabetes is presented in the scheme that is very complicated due to the structure and the treatment, which in this case is only one, it's insulin. And we also aim to describe an approach to define other types of treatment, but aim for the pancreatic cells that we can study in future for more further details. So thank you.
Thank you. Sorry that we missed you earlier.
Sorry, and apologies for my delay. I have personal issues to attend, but here I am. Thank you.
Oh, we're happy to have you here. So now let's continue back on with some more questions.
All right. We have a question for Jordi Cabelos. Does the model take into account the bus displacement or any other type of travel?
I think that Jordi might be a POTA presenter, so I don't know that he's here right now.
Maybe this question could be directed to Carlos Bustento.
Yeah, can you repeat the question, please?
Yeah, go ahead, Anush.
Well, can you do the movement or mobility estimates that you are doing? Can you use different sets of data other than bus transportation data, for example?
Oh, yes, I guess, but bus transportation data was the only available source that we had at the moment. But we can try to explore any other source.
So like, for example, do you have any idea what we can use, like if it is available? Like a cell phone data, probably?
Yeah, that's a good idea. I haven't thought about it, but yeah, it's a good idea to use that, yeah.
Thank you. OJ.
All right, thank you, Anush. There is a question for Paul Hurtado, but before Paul, you answer the question, I'll make a quick comment. The question to Paul is, could you provide us with references and what's the motivation for this? I believe providing with references will require posting things perhaps in the chat, or my suggestion would be the back of the conference program has emails. So if you're interested in references or more information from any other author, you might like to contact them directly. Having said that, could you answer the second part of the question? What was the motivation for your work?
Yeah, sure. Hey girls, could you whisper real quick? Thank you. Sorry, kids. Yeah, so I put a link in the chat there. And if you go to my webpage on my research description, there's a little blurb about this generalized linear chain trick stuff. And at the bottom, there's a link to a published paper and 3 preprints, and you can get more details from those. So as far as the motivation for it, the motivation was really to, well, let's see, it's complicated, but I'll try and make it short. I had a student and we were thinking about taking some models that had delays built into them and asking whether or not it mattered if you used a sort of linear chain trick type ODE model delay, or if you used a hard delay differential equation T minus tau type delay, or if you used a distributed delay like integral differential equations. And our thought, and I still think this would be a really cool little recipe for cranking out some cool papers, was to take some models from the literature, make those 3 different versions of that model, and then go through and compare the dynamics of those models to get a sense of how those different delays affected the results, right? And so in doing this, we found a really nice paper, a couple of papers actually by Jilan Feng and her group, who were looking at modeling Ebola and sort of hospitalizations. And there's this quirk in the way that we would normally write down by rule of thumb an ODE model that has infected individuals going to the hospital, which is that there's this like Markov property where as soon as you go to the hospital, whether you've been sick for 5 minutes or you've been sick for 2 weeks, right? Everybody's got the same remaining distribution of time spent in the hospital that they have to go through. And that doesn't really make sense, right? And so they went through and showed that if you sort of rethink these distributional assumptions in terms of how long individuals stay in different states, and in some cases, whether or not you can make those sort of conditioned upon time already spent in certain states, you can do a better job of deriving ODE models that don't have these sort of quirky assumptions built into them. And the way that they did that was the sort of formal, write down some integral equations, differentiate those using the sort of more formal linear chain trick, and it's a mess.
Right.
If you don't like working with integral equations, it's a mess. If you do, it's standard stuff, right? But there's a whole lot of people in the world who don't like working with integral equations that can write down ODE models. And so the real motivation was to go through and make it simple. And so that sort of led us on this.
All right. Thank you so much. Thank you. Brandilyn, the same question also goes to you. If you could provide the references later for your talk, either post them on the chat or I'll ask the question proposer to contact you directly. Carla Martinez, we have a question for you. Does your model take into account the effect of people that might go to Tijuana from other parts of the country? That's to Carla Martinez. Perhaps we lost her.
Carla is not here. She was she presented a poster.
Okay. All right. Great.
But no, the model does not take into account that kind of population.
All right.
Only what the website on the Secretary of Health tells us.
All right. Thank you so much. Panukaj Golati, I assume you're here. Can your approach be applied to Petri nets or finite state automata? I have too many windows in front of me, so I can't see.
I don't know that they're here with us.
Yeah, if
right
they
now.
are here or not. Brandolin, I have a question. This time you need to answer it.
I'm ready.
All right. So what was your motivation in pursuing your interest on ascertaining the maximum number of models in a data set?
Yes, thank you for that question. I did answer it in the chat, and I will just summarize that. So the idea is that we notice that some data sets, I'm always interested in reverse engineering. So starting with data and then building models that fit the data. And so the general problem is that you have too many models which fit the same data set. So we notice that some data sets give rise to many, many models, but other data sets, same number of points, but just different data, will give you a few data points or a few models. So then mathematically, what we were interested in was, are there data sets, or can we characterize data sets that give rise to a unique model? And then from like a modeling perspective, that may be hard to find. So, what we're looking for is ways of providing guidance for experimentalists in conducting their experiments, keeping in mind a modeling view, like which data sets are likely to give you just a few models that you can actually test in the laboratory, because it's not helpful having some data that you collect that give you, tens or hundreds of models. That's not helpful. But if you can design your experiments well to where you just have a few models that then you can actually test them, and it makes for much better, it enhances the predictive power of the models that you do generate.
Thank you, Brandolin. Sharon, we have a question for you.
Yep.
All right. Is your tool publicly available?
Yes, and I think I posted a link to the website in the chat. At first I did it just to the panelists, but I think I then did it to the panelists and all attendees. So yes, it's hosted by, I should have said, rShiny, and they are generously hosting it for free. So
perfect. Great.
Yeah.
Can you post it again, please?
Debasmita, I hope you're here. Do you use any special constitutive laws for the biological soft tissue?
Yeah, so I haven't used that till now, but in future I will be trying that. So far I have only considered level of concentration of the components involved in the atherosclerotic plaque formation.
Thank you. And we have only just a couple of questions left, but also a couple of minutes left. So I'll ask the next two presenters to take attention to the time restriction. Prachi, are you able to observe competition black-white strains in your model?
Yeah, at the moment, the way the model is set up, probably not, unless we were to specifically just simplify it down to, if the different strains have different probabilities of mutation. But yeah, the competition between different strains is definitely an interesting thing to look into.
All right, thanks. And one more question to Richard Clark and Darwin. Is your model with discrete phases or as a continuous function over time?
We answered this in chat briefly,
Okay.
but it's a straightforward answer. It's continuous over time.
All right, okay. And then last but not least, our favorite attendee, Glenn. In case you have not answered this in the chat, when you created your models, when you constructed your models, did you know or suspect which one was true before you picked them?
I don't know what you mean by which one was true.
I
think we're talking about the news, or if.
Okay. Which of the public statements?
Yes.
I would guess that most mathematical epidemiologists knew immediately when they heard those public statements whether they were true or not.
Okay, fair enough. And so this concludes our question answer session. Now, I'd like to introduce Dr. Joanna Wears from University of Richmond. She is the organizer of the next year's 2021 Beer Symposium. So I'll let her tell us a little bit about the next beer. Joanna?
Thank you, Olcay. It's been really interesting hearing about all of your work. This has been such a fabulous beer conference, and I'm hoping the next one will be too. And hopefully we'll be live in Richmond. That's the hope. So we'll be in person on our campus at the University of Richmond in Virginia. If you've never been to Richmond, Virginia, we have a lot of history here, as well as a river that runs through the city. So it's a beautiful place to come. There's a lot to see. We're 2 hours south driving from DC, and we're near North Carolina, and there are lots of things nearby if you want to come. And so I hope you will all be there. It's going to be a really fabulous conference, hopefully in person.
Thank you. And
do you have dates for that at this point?
No, we only know that it's gonna be in October. But beer, so well, I guess yes and no. Generally, beer always happens during the Columbus Day weekend, which is the first Monday of October. So whatever is the first weekend just before that is perhaps the tentative dates right now.
Hi, I got a question. For us, I mean, at least for my colleague and for me, it's difficult to go to Virginia. But I don't know if there's a plan to make also a room, a virtual room, in order to have this type of conversation and all this talk about some issues that were not very clear on the presentation. But I don't know if you had a plan also to make this organization in a virtual way, in order that we can also attend to this event in the 2021.
The answer is kinda, sorta, yes. So there are actually plans now to hold a starting 2022. In 2022, Beer Conference will be in England in July. And after that, we are actually planning to host 2 beers a year, one in the summer for in-person beer, and one in November, e-beer like this. So I guess there are plans, there are considerations taking place as we speak.
And Olcay, I think we can definitely think about even in the in-person, Diana, having
an online session.
Telepresence setup. I think that's a great idea. I think
within the beer and online session for those who can't make it. Yeah. All right.
Especially this last part, which was so great, hearing these quick summaries and where to find interesting research. That's a really great idea.
All right, great. So it's my honor to invite Dr. Padu Sesashiar for his closing remarks.
Oh, I wasn't planning for that, but because this is a great session, and it's, I mean, it's fantastic. And on a Sunday, you're all able to come and summarize. And given that this was a COVID impacted year, the fact that we expected you all to submit a video, you all fearlessly submitted really rich videos, but I think this session helped us to summarize what you all did. And so now even those that did not watch the videos will be tempted to go watch the videos now. So I think this is a great way to actually learn. So I think this format is the first time we have tried this out, and it's actually worked out really well. And hopefully we'll also add another part where you all start to talk to each other also. That was another part of the plan, to have a collaborative session. So congratulations to all the talks. It was fantastic. Thank you very much. Thanks, Olcay.
Thank you all, and have a safe trip back home.
Ojay, if I may add one final thing. Well, again, as Padu mentioned, congratulations to all of fantastic. This is the first time we tried these type of things, and we welcome any suggestions, comments, as you guys are giving in the chat message, for what else we can do. And just to add that we are continuously also organizing a webinar series. And keep an eye on the Intercollegiate Biomathematics Alliance website, where we kind of post all these things. We have a social media account on Twitter. It is called IBA Tweets, as well as it is called Letter in Biomathematics, which is a journal that we run through IBA, Intercollegiate Biomathematics Alliance. So keep an eye on that. Become friends on the Twitter so that you are aware of all what we are doing. And that way you can see many other activities and participate in it. This year, definitely we are having all these things as virtual. So just to add this point. OJ.
All right. Once again, we concluded one more beer conference without taking a gap year. I am all looking forward to seeing you next year in Richmond, if not before on other events. Thank you all and goodbye.